Analysis of macromolecular structure, Ramachandran plot. Leading objective, protein structure classification, confirmation of a protein. Understanding the Ramachandran plot, validation of a protein structure using Ramachandran plot. A role of proteins in biology. The proteins are functional tool of living organism, plays vital role in cell functions such as structural support, defense against pathogens, transportation along the cell walls, etc. Proteins are composed of 20 different amino acids with a different chemical composition of nitrogen, carbon, hydrogen, and oxygen. The function of each protein directly relates to the structure of the protein. To attain particular structure, proteins were synthesized using 20 different amino acids having different chemical structures and characteristics. This function of a protein varies with structure, varies with the composition of the amino acids and the sequence complexity. Protein structure classification. Structure of a protein can be classified into 4 classes: primary, secondary, tertiary, and quaternary structures by degree of structural complexity. Primary structure is amino acid sequence that creates polypeptide chain. In secondary structure, non-covalent interactions, particularly hydrogen bonds between amino acids, form preliminary 3-dimensional structures such as alpha helices and beta strands. Tertiary structure describes assembly of secondary structure to obtain the overall structure of the protein, mainly divided into globular or fibrous proteins. Quaternary structure explains the 3-dimensional structure representation of a protein having 2 or more polypeptide chains linked by disulfide bridges or hydrogen bonds. Conformation of a protein. The spatial arrangement of atoms in a given molecule is described as conformation, which is true also for proteins. Conformation explains not only the spatial arrangement but also the possibility of assuming other possible orientations. This change is not via breaking covalent bond but mere rotating a bond that connects atom together. This conformation changeability of a protein helps for exhibiting a function such as like on-off switch to function or adaptability upon approach of a ligand, which may be a small or macromolecule. The stable folded functional conformations are called native protein. The studies and conformation of a protein give the understanding towards the functional mechanism of a protein. The X-ray diffraction experiment used to get the possible stable conformation of a protein. From the diffraction experiment, researchers used to build protein structures. Ramachandran plot or 5 psi plots serving as a tool to validate the arrived model from any experimental technique and remained nearly unchanged in the past 15 years and continue to be an integral part of structural biology. From the above figure, one can understand that the phi and psi are torsional angles about n is equal to C alpha and C alpha C bonds by which the two adjacent peptide units can be oriented. So this one is the figure. Phi angle can be seen and the psi angle can be seen. Omega will be there. Ramachandran protein structure refinement. Since structure determination by X-ray crystallography about 8000 PDB entries and NMR become routine, usage of this plot is also increased to access the quality of the structure. These are the frequent questions put forward to a crystallographer. Is the crystallographic model position of atoms is justified? Since the enzymes or proteins perform functions by dynamic motion of atoms, does crystallography give us any insight into these motions? Are some part of the molecule are flexible than others? Are major movements of structural elements essential to the molecule's action? And the last 3 questions can be answered from thermal factor analysis for individual amino acids. The first question demands the fundamental validation. How does Ramachandran plot validate the protein conformation? Steric clashes. In space, at any instance, atoms cannot occupy same space. The covalent bond cannot be broken without any energy transformation. This possibility of rotation and clashes may occur between atoms, and only 2 possible degrees of rotation in amide backbone. From the above knowledge, Professor G. N. Ramachandran made 3 strategies to find steric clashes in mono and dipeptide. Phi is near 0 degrees Celsius, O1 OH clash. Psi is near 180 degree, O1 O2 clash. At any value of psi, O1 O2 clashes are observed at 0 degrees Celsius is equal to minus 45 to 45. So phi is near 0 degree Celsius. 0 degree. Next strategy is the backbone side chain clashes. To search clashes between side chain and backbone, the psi was varied and it found to be a steric clash between RH2 from the range of minus 65 degree to minus 175 degree. Overall backbone side chain interaction by varying psi and phi. The Ramachandran plot. From the above findings, Professor Ramachandran developed the following plot to describe allowed psi and phi combinations between amide planes. Anti-parallel sheets. When we draw this, anti-parallel sheets will be there. Anti-parallel beta sheets, parallel beta sheets, collagen triple helix, right twisted beta sheets, left-handed helix, although possible, not observed, right-handed helix, decrease and degrees. Axis will be psi angle and phi angle and degrees. Anti-parallel beta sheets, parallel beta sheets, collagen triple helix, right twisted beta sheets, left-handed alpha helix, right-handed alpha helix. From the analysis of existing crystal structures available at that time, the preference of psi-phi combination by secondary structure components were assigned. Unique features of Ramachandran plot in structural analysis. The Ramachandran plot clearly depicts how well the phi and psi angles cluster and reveals other oddities that may be the result of errors made during refinement. Ramachandran plot is one of the simplest, most sensitive means for assessing the quality of a protein model in the absence of experimental data, since the psi and phi angles or combinations of this are not usually retained during extra refinements as opposed to bond angles and bond angles. For instance, crystallographic software packages and map fitting programs usually contain a route for computing psi and phi for each residue from the current coordinate list, as well as for generating the Ramachandran diagram and plotting a symbol of residue number at the position. Ramachandran plots are dynamic when you change the molecule. Ramachandran plot get updated to reflect these changes. 5 probability density changes according to the selected residue type. There are 3 different residue types: GLI, PRO, and not GLI or PRO square. When you mouse over a representation of a residue, a little square or a triangle, the residue label pops up. The residue is active. It can be clicked. The graphics view changes so that C alpha of the selected residue is centered in the Ramachandran plot window. The current residue is highlighted by a green square. Some good things about outliers. When an outlier wants some discrepancy, sometimes there could be a reason for such anomaly. In fact, such disagreement may occur as a response to an input signal such as ligand binding. As a consequence, such conformation change could contribute changes in enzyme activity or in the affinity of protein ligand, protein or protein DNA interactions. Hence, structure determines the function of a protein. To arrive a correct model of the structure is very important. Ramachandran plot is highly useful to validate an experimentally determined structure. The plot explains the possible orientation energetically allowed to two successive peptides until with relevant to central C alpha carbon atom. Apart from this, the plot is highly useful to monitor the progress of a successful structure refinement. Hence, in all the structural refinement software, a prot is incorporated to see the real-time movement of psi and phi combination upon changes in orientation of a given amino acid. The unavoidable outlier in a plot may contribute for the function of a ligand or any exhibiting a molecule as such kind of movement arrangement. A Ramachandran plot, also known as a Ramachandran diagram, was originally developed in 1963 by G. N. Ramachandran, is a way to visualize backbone dihedral angles against OP of amino acid residues in protein structure. Different secondary structure elements have their characteristic torsion angles, which can be visualized using the Ramachandran plot. In a polypeptide, the main chain NC alpha and C alpha C bonds relatively are free to rotate. These rotations are represented by the torsion angles phi and psi respectively. G. N. Ramachandran used computer models of small polypeptides to systematically vary phi and psi with the objective of finding stable conformations. For each conformation, the structure was examined for close contacts between atoms. Atoms were treated as hard spheres with dimensions corresponding to their van der Waals radii. Therefore, phi and psi angles which cause spheres to collide correspond to sterically disallowed conformations of the polypeptide backbone. In case where the portion of protein X-ray structure was not properly defined, and even for bad or wrong homology models, we may find torsion angles in disallowed regions of the Ramachandran plot. Usually, these deviations indicate problems with this structure, suggesting that Ramachandran plot may be used in assessing the quality of experimental structures or structures built using homology modeling. The Ramachandran plot provides valuable information on the quality of a protein structure and gives some additional insight into the relationship between amino acid sequence and the three-dimensional protein structure. A Ramachandran plot can be used in two somewhat different ways. One is to show a theory which values or confirmation of the angles are possible for an amino acid residue in a protein. Second is to show the empirical distribution of data points observed in a single structure in usage for structural validation or else in a database of many structures. So Ramachandran plot is there. So the top region contain beta sheets. Left-handed alpha helix will be there and right-handed alpha helix will be there. Beta sheet, left-handed alpha helix and right-handed alpha helix. 180 0 psi 180. Beta sheet, left-handed alpha helix and right-handed alpha helix. Different secondary structural elements have their characteristic torsion angles which can be visualized using the Ramachandran plot. Torsion angles. The backbone or main chain of a protein refers to the atoms that participate in peptide bond ignoring the side chains of the amino acid. The conformation of the backbone can therefore be described by the torsion angles, also called dihedral angles or rotation angles, around the C alpha N bond and the C alpha C bond. C alpha N bond is called psi bond and C alpha C bond is called phi bond of each residue. Torsion angles, which are a function of 4 atoms, are considered to be variable and ranges from 0 to 360 degree. Typical values for psi and phi are -72 and -74 for an alpha helix and -19 and 113 for beta sheet respectively. The allowed ranges of psi and phi for an 80-degree fall into a defined region in a graph called Ramachandran plot. The uses: one is to show in theory which values or conformations of the psi and phi angles are possible for an amino acid residue in a protein. A second is for structure validation. Each dot in the Ramachandran plot shows the phi and psi values of an amino acid in the protein. The conformational freedom and therefore the torsional angles of a polypeptide backbone are sterically constrained. For some torsion angles, the atoms within the polypeptide chain will come too close to each other, a so-called steric clash. Rotation around the C alpha N and C alpha C bonds to form certain combinations of psi and phi angles may cause the amide hydrogen, the carbonyl oxygen, or the constituents of C alpha adjacent residues to collide. Steric interferences between adjacent peptide groups. Rotation can result in a conformation in which the amide hydrogen of one residue and the carbonyl oxygen of the next are closer than their van der Waals distance. The Ramachandran plot shows these regions of phi and psi where there are no steric conflict. On the Ramachandran plot, steric conflict regions can be easily distinguished. For a high quality experimental structure, these regions are simply empty or almost empty. Very few amino acid residues in a protein have their torsion angles within these regions. Beta sheets here, left-handed alpha helix will be there and right-handed alpha helix will be there. So in this picture, white areas represent regions which are sterically disallowed for all amino acid except for glycine, which is unique in that it lacks a side chain. All white portion are the places which are disallowed for other amino acids. Red region, conformation where there is no steric clashes. Allowed regions, red regions are allowed regions. Yellow areas are show the allowed regions. Slightly shorter van der Waals radii are used in the calculation. Atoms are allowed to come a little closer together. The conformation of most amino acids fall either between the alpha R or beta regions. Glycine has access to additional conformations. In particular, it can form a left-handed helix. The allowed regions generate standard conformations. A stretch of consecutive residues in a conformation generates an alpha helix. Repeating the beta conformation generates an extended beta strand. Two or more beta strands can interact laterally to form beta sheets. Thus, the Ramachandran plot provides valuable information on the quality of a protein structure and gives some additional insight into the relationship between the amino acid sequence and the three-dimensional protein structure. G and Ramachandran used computer models of small polypeptides to systematically vary phi and psi angles with the objective of finding stable conformations. For each conformation, the structure was examined for close contacts between atoms. Atoms were treated as hard spheres with dimensions corresponding to their van der Waals radius. Therefore, psi and phi angles, which cause spheres to collide, correspond to sterically disallowed conformations of the polypeptide backbone. Torsion angles, which are a function of four atoms, are considered to be variable and ranges from 0 to 360 degree. The three principal dihedral angles are commonly referred to as the omega, psi and phi. Together they define the protein backbone for each amino acid in a polypeptide chain. Rotation is permitted around the N C alpha and C alpha C single bonds of all residues. The angle around these bonds and the angle of rotation around the peptide bond define the conformation of a residue. Typical values for psi and phi are minus 72 and minus 47 for an alpha helix and minus 119 and 113 for a beta sheet respectively. The plot was developed in 1963 by G and Ramachandran et al. By plotting the psi values on the x-axis and the phi values on the y-axis. Plotting the torsional angles in this way graphically shows which combination of angles are possible. The torsional angles of each residue in a peptide Define the geometry of its attachment to its two adjacent residues by positioning its planar peptide bond relative to the two adjacent planar peptide bonds, whereby the torsional angles determine the conformation of the residues and the peptide. Many of the angle combinations and therefore the conformations of residues are not possible because of steric hindrance. By making a Ramachandran plot, protein structure scientists can determine which torsional angles are permitted and can obtain insight into the structure of peptide. The figure above shows the three main chain torsion angles of a polypeptide. They are phi, psi, and omega. The principle that two atoms cannot occupy the same space limits the value of conformational angles. The allowed ranges of and 4 is equal to 180 degree fall into defined regions in a graph called Ramachandran plot. The peptide bond has partial double bond character, forcing the OCNH atoms of the polypeptide backbone to be planar. Thus, only degree of freedom for rotation in the polypeptide backbone are around the bonds to the alpha C carbon phi or psi. However, there are significant limitations as to which angles of psi and phi can be used due to steric clashes between atoms. For some torsion angles, the atoms within the polypeptide chain will come too close to each other, a so-called steric clash. The Ramachandran plot shows those regions of psi and phi where there is no steric conflict. White areas correspond to regions which are sterically disallowed for all amino acids except glycine, which is unique in that it lacks side chain. The red region corresponds to corresponding conformation where there is no steric clashes. They are allowed regions. So beta sheet, left-handed alpha helix, right-handed alpha helix. The yellow areas are allowed regions. Slightly shorter van der Waals radii are used in calculation. Atoms are allowed to come to a little closer together. The conformation of most amino acids falls into either the R of regions. Glycine has access to additional conformations. In particular, it can form a left-handed helix. L-glycine has no side chain and therefore can adopt phi and psi angles in all four quarters of the Ramachandran plot. Hence, it frequently occurs in turn regions of proteins where any other residue would be sterically hindered. The allowed regions generate standard conformations. A stretch of consecutive residues in the alpha conformation generates an alpha helix. Repeating the beta conformation generates an extended beta sheets. Two or more beta sheets can interact literally to form beta sheets. The Ramachandran plot and the quality of a protein structure. In cases when the protein X-ray structure was not properly refined, and even for bad or wrong homology models, we may find torsion angles in disallowed regions of the Ramachandran plot. Usually, these deviations indicate problems with the structure, suggesting that Ramachandran plot may be used in assessing the quality of experimental structures or structures built using homology modeling. The figure below show how Ramachandran plot for the same protein structure. Red region indicate low energy variations, brown allowed regions, yellow the so-called generously allowed regions, and a pale yellow marks disallowed regions. The structure used to generate the plot on the left is 2.9 angstrom and was not properly refined. The structure used to generate the plot on the right is more recent and was refined against 1.8 angstrom resolution X-ray data. You may notice that the torsion angles on the left plot lack real clustering around secondary structure regions and show a much wider distribution compared with the plot on the right. There are also many dots in the disallowed regions in the red plot and almost none in the right. Thus, the Ramachandran plot provides valuable information on the quality of a protein structure and gives some additional insight into the relationship between the amino acid sequence and the three-dimensional protein structure. A Ramachandran plot can be used in two somewhat different ways. One is to show the theory in which values are used, or confirmation of the psi and phi angles are possible for an amino acid residues in a protein. Second is to show the empirical distribution of data points observed in a single structure in usage for structure validation, or else in a database of many structures. Either case, it's usually shown against outline for the theoretically favored regions. The first Ramachandran plot was calculated just after the first protein structure at atomic resolution was determined. It is the myoglobin in 1960, although the conclusions were based on small molecule crystallography of short peptides. Now, many decades later, there are tens of thousands of high-resolution protein structures determined by X-ray crystallography and deposited in the protein data bank. Many studies have been taken advantage of this data to provide more detailed and accurate psi and phi plots.